virus bind to the cell surface by means of these receptors on their surface, and after binding, what happens? The cell virus is internalized via receptor-mediated endocytosis. Eventually, it is able to release its DNA into the host cell cytoplasm, which take hold of the host cell. But the host body is also able to produce some antibodies which binds to these receptors on the surface. As a result, the virus is not able to bind to the cell surface of the host and cause infection. IgG provides immunity to viruses by neutralization in tissues, and IgA provides neutralization of viruses in cell linings. For an exotoxin to cause harm, it must first bind to the receptors on a susceptible host cell, which results in endocytosis of the host receptor complex inside the host cell, where the dissociation of the toxin results in release of active chain, which is poisonous to the host cell. So the host cell produces antitoxin antibodies, which binds to the toxin via their fat portion and neutralize them. So now the toxins are not free to bind to the receptors of the susceptible host cells. So IgG and IgA both provides immunity to toxins via neutralization. IgE binds to the surface of mast cells and vesicles and promote inflammatory response. These IgE are bounded to the surface of the mast cells, the vesicles, by means of their Fc region. However, their Fab region is free. Whenever they recognize an antigen or an allergen, this Fab portion binds to that antigen and results in triggering a series of signals, which results in release of inflammatory mediators by the mast cells or the vesicles. The resulting inflammatory response subsequently delivers defense cells and defense molecules from the bloodstream to the site of infection, as we have studied in the process of inflammation.
An allergy is an overreaction of the immune system to a normally harmless substance called an allergen. Common allergens include pollen, animal dander, down feathers, mites, chemicals, and a variety of foods. On first exposure, the inhaled allergen enters the mucous membrane lining the nasal passages, where it is taken up by the antigen-presenting cell, which presents it to the T cells. These T cells activate the B cells to release substances called IgE antibodies against the allergen. These IgE antibodies sit on the surface of the mast cells. The mast cells have granules containing chemical mediators like histamine and prostaglandins, etc. On exposure, the allergen binds to the IgE antibodies present on the mast cells, cross-linking them. This results in the release of histamine, prostaglandins, and other mediators into the surrounding tissue. These mediators cause dilation of the surrounding blood vessels and increase their permeability. This results in the nasal stuffiness, sneezing, and mucus discharge of allergic rhinitis. Antihistamines work by blocking the action of histamines at its receptors and thus decreasing the body's reaction to the allergen.
It's time for our second concept checkpoint. In the first question, you have to find out whether viruses can resist neutralization by antibodies. In either case, you have to justify your answer. In the second question, you have to find out the type of class and the role of Fab and Fc region in antibody-dependent cytotoxicity and the process of neutralization of viruses by antigen-antibody interaction. Try to find out the answers and we will discuss them in our question answer section.